accomplished by the annulus that encircles the sporangium. The annulus begins to dry out as water evaporates from its surface. The tension or pull between the remaining water molecules and the wall now increases. The thick inner and side walls resist this, but the thin outer walls are easily pulled inward. Pulling in the outer walls causes the annulus to contract much like an accordion. Shortening the annulus tears the lip cells apart. As the annulus continues to shorten, the tear enlarges and the spore case opens further. When too much water is lost, the water molecules are no longer able to hold together. When this happens, the pull on the wall is released and the annulus springs forward, closing the sporangium so fast that the spores are thrown out. This process is repeated in thousands of sporangia on a leaf, so that large quantities of spores are released. When spores land on a moist surface, those which germinate first will form bisexual or hermaphroditic individuals. A rhizoid emerges and attaches the spore to the soil. This is followed by a sheet of cells, which is the young gametophyte or prothallus. A notch forms on the prothallus that contains dividing cells. This is called the notch meristem. Continued growth results in a heart-shaped bisexual gametophyte. The lower surface is firmly attached to the soil by numerous rhizoids. Male gametangia, called antheridia, are formed at the posterior end of the gametophyte. The outer wall of an antheridium consists of ring cells and a cap cell, which surround fertile tissue. At the anterior end of the prothallus, close to the notch, there are female gametangia called archegonia. An archegonium consists of a neck containing a neck canal cell. At the base of the neck, there is a swollen region called the venter that contains an egg cell. Gametangia of both sexes may be present at the same time or at different times. The timing of their appearance will determine if there will be self-fertilization or cross-fertilization. In some ferns, archegonia are formed first, and when flooded with water, a hormone called antheridiogen is released. This hormone will stimulate adjacent plants to stop growing and form antheridia, but no archegonia. A bisexual gametophyte in a female phase may therefore be surrounded by several male gametophytes, increasing the chances of cross-fertilization. Being close to the soil, the small gametophytes are easily flooded when it rains. This water plays an important role in fertilization. Water stimulates the cap cell on the antheridium to open, releasing the sperm cells. The flagellated sperm are now able to move within the water in search of an egg. Sperm consist of a spiral cell body bearing numerous flagella that move it forward. Water also stimulates the archegonium to open. The contents of the neck canal then diffuse into the surrounding water, where they act as a sperm attractant. The attractant stimulates a sperm to swim towards the open archegonium. It then swims into the opening and moves up the neck canal towards the egg cell. Fertilization is accomplished when the egg and sperm nuclei fuse. This creates a single diploid cell, the zygote. The fertilized egg remains attached to the gametophyte. Inside the archegonium on the lower surface of the gametophyte, the zygote begins to divide, forming the embryo's sporophyte plant. The uppermost part of the embryo, the foot, absorbs nutrients from the surrounding gametophyte tissue. As the embryo grows, the venter tissue stretches and eventually ruptures. A root now grows into the soil for support and to absorb water. Other parts of the embryo form the first leaf and the future stem, the rhizome. The leaf grows through the notch in the gametophyte to reach the sunlight. Eventually, the rhizome emerges and provides additional leaves and roots. In this way, a new plant is formed that matures into another spore-producing fern, completing the reproductive cycle. We have seen that the fern reproductive cycle contains two plants, the large familiar spore plant or sporophyte, and a tiny gamete plant or gametophyte. Using spores to travel great distances and gametes which introduce genetic variability, ferns have spread and evolved throughout the world. In cool, temperate regions of the world, there are forests of cone-bearing trees known as conifers. One group in particular, the pines, is especially familiar and comprises one of the most extensively traded crops. Pollen plays an important role in pine reproduction, and we know that it's released from small cones called pollen cones. The pollen cone consists of many small scale-like parts arranged in numerous spirals. Each scale in a single spiral is called a sporophyll and is attached to the central cone axis. On the underside of each sporophyll, there are two microsporangia. An outer epidermis covers the sporophyll and its sporangia. Beneath the subepidermal tissue, there is a layer of cells known as the tapetum. The tapetum tissue contributes to the development of the pollen grains. Inside of the tapetum, there is a mass of spore-producing cells called microsporocytes. The nucleus of a sporocyte cell is diploid, containing 2 sets of chromosomes, one from each parent. As each sporocyte prepares to divide, it deposits a layer of inert callose. Callose effectively isolates the microsporocytes from one another. The nucleus inside each sporocyte divides by meiosis to produce 4 haploid daughter nuclei. Each nucleus becomes surrounded by a wall that begins to enlarge at 2 points. The resulting microspore cells remain attached to one another and are known as a tetrad. The wall outgrowths develop into enlarged, gas-filled structures called wings. The individual microspores now increase in size. At this stage, a microspore consists of 2 large expanded wings and a smaller dome-shaped portion. The dome-shaped portion contains one of the spore cells from the original tetrad, complete with haploid nucleus and surrounding cytoplasm. 3 successive mitotic divisions now take place. These create 2 small prothelial cells, 1 generative cell, and 1 tube cell. These cells represent the immature male gametophyte. This entire structure, consisting of the external wall and the male gametophyte, is known as the pollen grain. A final deposit of sporopollenin completes the formation of the pollen grains, and they are released when the microsporangium opens. The pollen grains are now easily dispersed by air currents. Seeds play an important role in the reproduction of pine, and we know that they're released from large woody cones. However, when these cones first appear in the springtime, they're small and fleshy. At this stage, they contain ovules and are called ovulate cones. The intact ovulate cone consists of numerous spirally arranged ovuliferous scales. Each ovuliferous scale has a small bract fused to its lower surface. The upper surface contains 2 ovules. Each ovule has an outer protective layer or integument. An opening in the integument, the micropyle, consists of a canal that terminates in 2 micropylar arms, each covered with a sticky secretion. Beneath the integument, there is a fleshy megasporangium containing a diploid megasporocyte cell. When the cone is ready to receive pollen, the ovuliferous scales separate from one another, exposing the ovules inside. Carried by air currents, some pollen grains are forced between the ovuliferous scales and fall down towards the ovules. Some of the pollen grains adhere to the sticky surface of the micropylar arms. The megasporangium now releases a fluid. This is called the pollination drop. As this fills the micropyle, it picks up the pollen grains. The pollination drop soon moves back up the tube, taking the pollen grains to the surface of the megasporangium. At this time, the micropylar arms wither effectively sealing the pollen grains inside the ovule. The scales of the cone now grow together, sealing the ovules inside a tough protective barrier. Inside the ovule on the surface of the megasporangium, the pollen grain begins to swell and form a tube-like growth.